Hello, my name is Stephanie Moore. I am the director of the DRSC in the Paramount Lab at Harvard Medical School. I will be introducing you in this demo video to the FlyPrimerBank online resource, a database of pre-computed primer designs for quantitative real-time PCR, which is commonly called qPCR. qPCR can be used to determine the level of mRNA expression of a given gene in a given sample relative to some control. qPCR is often done to compare mRNA levels in a control sample to a sample that has been genetically perturbed or subjected to some treatment. In addition, qPCR is often done to determine if a reagent, such as an RNAi reagent that is designed to knock down or reduce mRNA levels, is effective, and to what degree that RNAi reagent effectively knocks down levels of the mRNA target. FlyPrimerBank was specifically designed to support qPCR studies in the fruit fly Drosophila melanogaster. It was developed by a team led by our Director of Bioinformatics, Dr. Claire Yanhui Hu. FlyPrimerBank also represents a collaboration between the DRSC and a developer of the original PrimerBank resource for mammalian studies, Dr. Xiaowei Wang. The design of oligonucleotide primers for applications such as Sanger sequencing or PCR are well established and straightforward. In contrast, designing primers for qPCR can be more complex. FlyPrimerBank helps researchers planning qPCR projects by providing a database of pre-computed designs and supporting information that will guide your choice of primer pairs. One of the first things you might notice is that FlyPrimerBank, like most DRSC bioinformatics resources, supports searches using single gene or using a list of multiple genes, as can be inputted here, or a list of genes provided as a file. For the purposes of this demo, I'll keep things simple and search with two genes, the wingless gene and the ribosomal protein S6 kinase or S6K gene. As you can see, I opted to input the official fly-based gene symbols for these genes, WG and S6K. You could alternatively enter the full name or a fly-based FBGN ID number. As a first run, we will leave all of the default options as they are and run a search. In the output, you see a table of results that indicate various primers pairs for each of our search terms. In addition, I want to bring to your attention that you can download the results by clicking a button that appears either at the top and also at the bottom of the results table. Moving across a row, we can see that we first are presented with the search term that we entered, then an identifier for the primer pair, the specific sequence of the forward and reverse primer, and their predicted melting temperatures, and the predicted size of the resulting PCR product. We also see the official FlyBase FBGN ID number and symbol for the gene, and we see an indication of what fraction of isoforms of the gene are targeted. In the case of wingless, there is only one isoform, and it of course is targeted by these primer pairs. In the case of S6 kinase, there are two possible isoforms, and all of the primer pairs that are displayed are predicted to amplify both isoforms. We next see what region of the gene is targeted, and in this case, we're targeting coding sequence with the primers that are indicated. The next column shows whether or not this particular primer pair spans an exon boundary. The answer is yes for this pair, indicating that at the genomic level, there is an intron that intervenes somewhere between these two primers that would not be present, of course, in the messenger RNA. Finally, we can look at the last three columns. This next column shows whether or not there is overlap with particular types of RNAi reagents in public collections. What we first see is that there is potential overlap between the primers predicted here and DRSC reagents designed for cell-based RNAi studies. In addition, we see potential overlap with one of the public collections of in vivo RNAi reagents. We next view the FlyBase release that was used to compile the currently displayed information, and we see a link to an alignment. If we click on this link, we see an alignment of the predicted qPCR product, which is shown here in green, with an indication of the mRNA, a diagram of the mRNA, showing its CDS start and the coding stop, showing the entire extent of the messenger RNA, showing the indicated positions with these blue triangles of exon junctions. So this is where introns would normally intervene would be intervening in the genomic sequence. An advantage of choosing a primer that spans one of these boundaries is that you can be sure that a product of this length represents a messenger RNA, because a product that incorporates the intron from the genomic DNA template that could be a contaminant would be of a radically different size. In addition, we see the predicted PCR product at a sequence level in this view. I will next walk you through some of the options that FlyPrimerBank provides on the search page, so you can tailor a search for a specific project. In this section, the options include checking overlap with DRSC amplicons for cell-based RNAi or with any of a number of reagents from the TRIP, NIG Japan, or VDRC collections that are designed for in vivo RNAi knockdown. In addition, there is the option to click a box that would include expression data in the output file. If we rerun our search, we will see that although the table doesn't change, when we run a search including expression data option, the download results now indicate that in addition to seeing the table results, you'll also include information from the Modern Code project on expression of the gene. As I mentioned at the beginning, you have the option to input one or more genes or upload a file of gene names. You also have the option at FlyPrimerBank to input a sequence. So let's try a search with the sequence. When you upload a fragment of a transcript, for example, in this case, the first exon of the gene OATP58DA, the BLAST program will blast the input sequence against the sequence of all pre-computed primers and identify the best primer pairs that can be aligned perfectly on the input sequence using both forward and reverse primer. If any qualified primer pairs are found, the program will link back to the target gene and also bring back all of the other primer pairs for this gene. So in this example, it's indicated that we use BLAST as our input. We then see the identity of the primer pairs. We see the gene symbol that matched, in this case as we expected, and we see potential primer pairs. We also make it possible for you to provide feedback. If you find that a given primer pair from FlyPrimerBank does or does not work, please make us aware of that finding by clicking the Submit Feedback About Primers link at the right-hand side of the landing page. In addition, if you have a primer pair that you have shown works for qPCR in Drosophila and that is not yet included in the database, we can add that pair so that your findings can help the community. This can be done, again, by selecting Submit Feedback About Primers. There's a template form that you will ask to fill out and upload. You can reach information about FlyPrimerBank resource from either the help or documentation links on this page. When we go to the documentation page, we see a number of things that might be of help. A text description, a workflow, and the publication, including a downloadable file for that publication. I want to point out that if you're looking for qPCR primers for transposable element detection, on our documentation page, we also point you to a helpful published paper that can point you in the right direction for those types of primers. We encourage you to read our paper in order to learn about how the resource was developed and more about how you can use it. We also would ask that if your research using designs from FlyPrimerBank results in publication, that you would please cite our paper in your methods section or another appropriate section of the manuscript. Citations and acknowledgements are important to our ability to continue to get funding for maintenance of existing resources and for development of new ones. Returning now to the home page, I would finally like to invite you to visit the link up here called Tools to view our Tools Overview page. The Online Tools Overview presents the long list of resources that we have developed for identification of gene lists, for identification of orthologs, reagents, for data mining, and for many other processes. They include databases, data analysis support, and data sets. They also include tools that are relevant to multiple genetic model species in humans, in addition to fly-specific resources, such as FlyPrimerBank.